Okay, here we go. Who are these guys? Right, brothers. Well, we have got a lot to learn.
It's black and white, and they look. They're studying airplanes. Yeah, right.
There's nothing to do with like airplanes at all.
The Harvey.
There, that is Watson and Crick.
Oh,
yeah.
So I want to talk for just a second about the time period during which this picture was taken. We're talking early 50s, okay? Not long ago. When we think about this in terms of perspective, my parents were born when they figured out the structure of DNA. So when we start learning about these scientists, we're talking about people that are still alive. In contrast to when we were talking about Mendelian genetics, Mendel's past, right? Most of the research we talk about in this class is historical to the point of talking about Darwin, that was 150 years ago. We're learning about so much fundamental biology that it happened a very long time ago. Because at this point, we're exploring deeper and deeper and deeper. But when it gets to the DNA stuff, we learned so much about DNA in the last 50 years, truly. And then when we consider the Human Genome Project, the research that we're doing about GMOs, all of these things have come after the fact. Everything to do with biotechnology has been developed since this picture was taken. It's an impressive amount of advancement, scientific advancement, that's happened in a very, very, very short period of time. And so just to kind of understand that as I go through this is pretty recent science in the world of science as far as that's concerned. And it just really shows you how very, very quickly our understanding of biology has changed and just how broad of a science it is now that before not as much. So the most important thing for you to take out of today's lecture is going to be that top bullet point. It says, of all of nature's molecules, nucleic acids are unique in their ability to direct their own replication from monomers. So I want you to think about that for a second. We talked before about the structure of DNA molecule, that A's match with T's and C's match with G's, almost without question. And as a result, we talked also about how the DNA strand was running anti-parallel, so it's like the divided highway where one road goes this way and the other road goes this way. Well, if I pull the two apart, this strand over here has all the information necessary to produce this strand over here. That's super important, because then if I take my strands and I pull them apart, this one can produce its complement, and this one can produce its complement, because all of the information for both strands is stored in one. Okay. That's going to become a major thing. Hereditary information is encoded in the chemical language of DNA, in the A's and T's and C's and G's, and it gets reproduced in all the cells in your body. So when we talk about DNA replication, what we're talking about is synthesis. Think back to your cell cycle. G1, S, G2, M. S was synthesis. And we said, well, what's happening in synthesis? We're replicating our chromosomes. What did that mean? It means we're duplicating our DNA. And I said, we'll get to that later. Today's later. Okay, we're there. So today we're going to talk about how did we discover the structure of DNA, how does DNA replicate itself, and then we're going to end on how does DNA fit into your nucleus. Because each of your nuclei has about 4 centimeters of DNA in it. But the cell itself is tiny. So how do we make that happen? So that's what we're going to get through all today. So all of chapter 13. You have a mastering biology assignment that is on specifically chapter 13 to cover this. Do everybody a favor and don't put it off. Okay, make sure that you get to this weekend. A lot of vocab, a lot of ACEs, a lot of enzymes in today's lecture. Okay. The DNA itself directs your development. It directs your biochemical identity, your anatomical structure, your physiology, and to a certain extent your behavior. If you're taking psychology, or if you ever do take psychology, you'll talk about the genetic influence of behavior. It's there. It's very real. So 100 years ago, identifying the molecules of inheritance was a major challenge to biologists. And the reality is biologists, people responsible for studying all of life, had noticed some trends. They knew the genetic material was in the nucleus. And then they'd gone into the nucleus to see what was there, and they found two things. They found DNA, and they found protein. And they're looking at life, and they're looking at the diversity of life, and they're saying, wow, this is true for bacteria, and elephants, and algae, and plants. Everything on the planet that's alive, in that nucleus is DNA and protein. And we have a huge diversity of life on this planet. So then they look at these two molecules, and they see DNA. Very consistent structure, pretty uneventful molecule with 4 monomers. And then they look at proteins. Proteins that can take as many different shapes as you can imagine, that can be as small or as large, and 20 different amino acid monomers. And they're looking at the DNA molecule that seems very, very simple, and they're looking at the huge complexity of proteins, and they're looking at the huge variety of life on this planet, and they came to the conclusion that it must be protein. That is the heritable material. Oops. They couldn't prove it. Well, why couldn't they prove it? They were wrong. They could not show that protein was the thing. So, along came an accident, which is usually how most good science starts. What happened was this guy named Frederick Griffith was trying to develop a vaccine for pneumonia. And pneumonia was at this time frame something that people died from. And in fact, pneumonia is still something some people don't recover from, but it's not nearly as deadly as it once was. But what he found in an accident and then developed into an entire experiment was this. He took his pneumonia. He was trying to, the way vaccines work is that if you are able to survive something, then you have antibodies for it, and then we take those antibodies and we put them in other people, and then they don't get it also. So he was trying to get mice to live through pneumonia, essentially. But what happened was, he took some, I'm going to say 6 cells, it says S. It doesn't stand for sick, but it's fine. 6 cells, and he injected them in the mouse, and the mouse died. As expected, pneumonia will kill a mouse. Got it. He wanted to, though, show that just giving a mouse a shot doesn't kill it. I mean, maybe it's very stressful. So then he gave the mouse a shot of this stuff. Healthy cells. The mouth is like, cool. Then he takes some of these, boils them, killed them. So there are 6 cells that can't make you sick, and the mouse is fine. Typically, this is actually one of the approaches to making vaccine. It wasn't just an accident that he did that. But this is where things got weird. When he took this, the mouse lives through this experience, the mouse lives through this experience, we put them together, and the mouse dies. Then he does a little mouse autopsy, we call that a necropsy. Does a little mouse autopsy, and inside he finds this. This, which is what killed our first mouse. Well, that's not normal. And he called this transformation. It's like the sun, more than meets the eye, right? How did that happen? How did these two things go in and that come out? So he gave it this word. We have transformed those bacteria. We now have a definition for that. We changed the genotype and the phenotype of a cell due to the assimilation of external DNA by the cell. Somehow, external DNA gets in there. Of course, Griffith didn't know that that's what was happening, so he just gave us the word. We gave it a definition later, but that was very peculiar. So somebody took a look at that and was like, okay, so we can change the phenotype and the genotype. How do we investigate how this happens? We thought investigating this phenomenon is how we're going to actually be able to sort out this DNA protein business. So they decided to actually investigate bacteria. We love bacteria as a research organism because they're very small, they're cheap to keep, and they reproduce very quickly. Okay. So we get these bacteria, and we get the viruses that infect them. It turns out viruses are much more than the cold and the flu. Viruses can infect anything alive, including plants and including bacteria. So the bacteria have these viral enemies, and we call them bacteriophages. I hate that the viruses call it bacteriophage because people think it's a bacteria. It's not. Remember, the word phage means eater. Remember, phagocytosis, cellular eating? Okay, so bacteriophage means eater. Bacteriophage or bacteria eaters. Well, they're not really eating the bacteria. Here's my bacteria. Here are my bacteriophages, my viruses, and all that viruses are made out of are protein on the outside and DNA on the inside. We knew that at this point also. Viruses are non-living. It's very weird because they're just kind of this weird, almost living chemical. But one of the criteria for life is that you can reproduce on your own. And they can't. They have to hijack something that is alive in order to cause it to reproduce. We'll learn all about viruses in the spring. But in essence, that's what happens. So this is not a living thing. But they look like these very angry, large-headed spiders. And when the DNA hangs out up here, and when they find a victim, they literally squat and poop the DNA into them. That's what happens. And there it goes. Okay? You don't like that? You should never have to teach you that class, have you? I would. So, that's what bacteriophages do. Okay? So we have our bacteriophages. We know they infect cells, bacterial cells. So Hershey and Chase came along, and Hershey and Chase said, okay, I can be smart about this. I'm going to create a whole bunch of viruses, and in half of them, I'm going to use heavy sulfur, okay, sulfur right here, sulfur goes into proteins, so that I can track where the protein is. And in the other half, I'm going to have heavy phosphorus. And phosphorus is present only in that sugar phosphate backbone, not in the protein. So by growing half of my viruses in heavy sulfur, I'm going to have labeled protein, and half of my viruses, I'm going to have labeled phosphorus, I'm going to have labeled DNA. I'm going to take all of my special viruses, and I'm going to let them infect bacteria, because that's what they do. And then after I've given them enough time to infect the bacteria, I'm going to see where my labeled compounds are. So when we labeled the protein up here, after we give the viruses enough time to infect those bacteria, we throw all the cells into the blender, an important lab tool, and break it all apart, spin it down in a centrifuge so that the heavy stuff, the bacteria, goes to the bottom. Look at the bacteria. Sure enough, the infected bacteria do not have any of this radio-labeled stuff. All of our labeled sulfur is floating around up here in our juice. So that didn't actually cause a change to the bacteria. But then when we look at this one, same thing. Viruses come in, infect the bacteria, blend them up, look on the inside, and lo and behold, that's where you have labeled DNA. So what actually went into the cells? DNA. Aha! So that's how we learned that DNA was the genetic material. This did not go over well in the scientific community, as you might imagine, because what we just found out is we just spent 50 years researching proteins, and that's kind of a loss, right? A lot of people's life work was suddenly useless, okay? So that kind of goes over like a lead balloon, right? Hershey and Chase had come out with this and said, nope, sorry guys, it is DNA. Critique it all you want, repeat the procedure all you want. Every single time you do this, this is what you're going to find. And sure enough, that's what happened. We knew almost nothing about DNA because remember, it was a pretty boring molecule that wasn't really worth our attention. Scientists knew that DNA was a polymer of nucleotides. We knew what nucleotides were, and we also knew that there were the 4 monomers. And those were the things that kind of caused us to be so dismissive of it in the first place. And so along comes this chemist, Chargaff. Chargaff was examining the DNA of lots of different species, mostly because he just thought it was fun. And he found 2 patterns that became known as Chargaff's rules. The base composition of DNA varies from species to species. Every species had a very different composition of A's and T's and C's and G's. But within a species, the numbers of A's and T's are roughly equal, and the number of C's and G's are roughly equal also. So Chargaff reported this. He thought it was very interesting. But nobody could figure out why that was the case. Explaining these rules was going to require an understanding of the structure of the DNA molecule. We knew what was there, but we didn't know how the thing was put together. And if we were going to make any advances in this area of genetics and hereditary issues, we'd have to figure out how this molecule was put together. Massive race to try to figure out how this was done. The answers they knew were all in the structure. James Watson traveled to England. James Watson was not a biologist. Traveled to England for some unrelated work. And while he was there, he ran into his friend Wilkins. He actually wasn't even working with Wilkins, is my understanding. And he wanted to go have lunch with him. They worked on something else previously. And Wilkins was like, oh, I have this super annoying lady down the hallway, and do you want to go see what she's got going on in her lab? Wilkins took Watson into Rosalind's lab to be nosy. While they were there, Rosalind was gone. She was out of the country. This picture was shown. Rosalind was working on this question of the structure of the DNA molecule. She was a biologist, trying to figure this out. Massive worldwide race to figure out who can figure out the structure of the DNA molecule. And Rosalind had taken hundreds of fancy pictures called X-ray crystallography diffraction images and could not get a crisp image. That was the only crisp image she could get. Hundreds and hundreds of these pictures. They knew that these were these massive molecules. They knew they were very long molecules. So they were expecting to see streams. But this was the only clear image she could get. And that wasn't a stream. She could not figure out what it was. But I told you Watson wasn't a biologist, right? He was more of a chemist. Watson did a lot of this technique. And so, as soon as he looked at that image, he knew exactly what it was. That's the image of the DNA molecule looking down the end, rather than looking at it from the top. Because remember, it's a double helix, right? It's like a phone cord, an old-fashioned one, when they were still attached to the wall. And so it's just like looking down the long, skinny end. But Rosalind couldn't figure that out because she was looking for something long and skinny and was trying to figure out how that was appearing here. But if you look down this image, you can see that it's spinning, right? That's what you're seeing. And then coming through the middle are going to be those bonds. Okay? She didn't know it. So what does Watson do? Something unkind. Watson doesn't say anything to Rosalind. Watson doesn't say anything to Wilkins either. Watson goes home to his friend Crick and says, hey man, want to win a Nobel Prize? So they immediately start doing some research on the structure of this molecule, reading up on what other people had published. He knew it was a spiral because he saw the picture. They had some available data. They knew the biochemistry of the monomers. This is all published stuff, right? This is science is free to anybody who wants to read about it. Observation of the helix in the photo, Chargaff's rules, the A's and T's and C's and G's, and then these X-ray measurements of the width and space between the bases. We knew that it was a long molecule that had these particular measurements. And here's what they saw. They saw that the entire molecule was the same width for the entire length. Remember when I made you learn purines and pyrimidines? Yeah, I know. Why? Well, so purines were the two-ring structures. Pyrimidines were the one-ring structures. But if you put two-ring structures next to each other, that makes it fat. Two one-ring structures next to each other makes it skinny. But if you pair up a 2 with a 1, it can be the same width the whole way through. Okay. So they looked at all of this, and they started to put together this concept. Well, what prevents this 2-ring structure, adenine, from binding to this 1-ring structure, cytosine? Why is it always going to be this one with this one? The answer was in the hydrogen bonds. Count them. 3 here, because we have nitrogen, nitrogen, oxygen. 2 here, because we have nitrogen, nitrogen. So this one can't be stable with this one, and vice versa. That's hugely important because remember that DNA is this long-lasting molecule that's supposed to last your lifetime. It's not like RNA, it kind of comes and goes as it pleases. Okay? This has to be very, very stable. So we have to have it balanced. So this is ultimately what they came up with. This was Watson and Crick's model. They hadn't yet figured out the 3' end and the 5' end. They're labeled in your textbook. But they figured out that we had these 2 anti-parallel backbones with the bases in the middle. And they knew that because they had seen that image running down the middle. So even though the base pairing rules dictated these very specific combinations, they did not restrict the sequence of the nucleotides. The A, T, C, G. The linear sequence of the 4 bases could be as varied as we needed. And each gene has a unique order or unique base sequence. So everything else we teach you about protein synthesis had to start here with our very basic, what is the molecule? So what happened next? Despite Rosalind Franklin having given the work, Rosalind Franklin had no idea that anything had even been looked at. She came back from being out of the country, the lab was as she left it, she continued her work. A few months later, the paper was published. She wasn't even mentioned. Watson and Crick, who wrote the paper, got Nobel Prizes for figuring it out. This is the worst part. Wilkins' contribution to this was unlocking a locked door. Also got a Nobel Prize for this. Why? Why did he do it? He didn't like Rosalind. It was the 50s. Where did women belong? Not in science labs. Watson didn't like the fact that she was there and contributing significantly. They were not friends. There was no love lost for sure. Rosalind died a few years later, 1958. She was only 38 years old when she died. She literally died giving her life for science. She died of radiation poisoning, actually, which wasn't too uncommon because we didn't understand cancer, because we didn't understand DNA. And she worked with these very dangerous molecules that gave her cancer and killed her. You cannot get a Nobel Prize posthumously. So at her best, she was alive for a few years after all this came out, she was acknowledged as a contributor. That's it. When it was actually her data the whole way around that actually allowed this to happen. So it's kind of sad. That's what a Nobel Prize looks like. I gotta be honest with you, if I ever win a Nobel Prize, I'm gonna expect it to look like something better than a penny. I mean, come on. I hope it has a fancy container. So Rosalind never got one. The other 3 guys did. That's a little sad, right? And it's kind of the way that history has treated women scientists up until really the last 30 or 40 years, when it's like, oh, equal. But when we get to the spring and we start voting on AP Bio t-shirts and putting your names all over them and stuff, I always provide one design and then you guys contribute your own designs also. And then we vote. And the one I design also it hasn't ever been printed, which is why we still do it. It has this picture on it, and it says "Long Live Rosalind" on the bottom of it. So you guys will prick something funny, I'm sure, because you guys tend to like something funny. But I try every year to always remember Rosalind and her contributions to genetics. And there are lots of kind of women scientists where this has been the case. So that's okay. It's a different social situation now, and women are now respected for their contributions in science, and we're grateful for that change. So the last line of Watson and Crick's paper read, "It has not escaped our notice that the specific pairing we have postulated immediately suggests a possible copying mechanism for the genetic material." So they were kind of snotty about the whole situation. Very much like, I told you they weren't biologists. And so they were like, oh, biologists, it's such a soft science, very dismissive. You can't solve your own problems, so we're going to come in as chemists and figure them out, and now we're going to go back to the real work of chemistry and leave you to your work. And that last sentence, I think, just very clearly articulates that. They're like, okay, we figured this much out for you, go on, go do the rest of your science, right? So one of them is still alive. I think Watson died and Crick is still alive, I can't remember exactly. To their credit, though, they have since apologized. They have since come out and been like, yeah, that probably should have been handled differently. And so I do appreciate kind of the hindsight being what it is. But once they published their paper, then it became, more of a race to figure out how does this work and what is it doing. And so as we move into this process of transcription and translation and gene expression from a molecular standpoint, all of that depended on this discovery. Right? We had to get this done first. So what Watson and Crick are suggesting is this semi-conservative model like I was telling you before. We have the parent molecule, it pulls apart, and we put in a copy of the daughter on either side. But as you might imagine, scientists were a little shy about making assumptions at this stage of the game because we had assumed protein was the important molecule and ran with that for 50 years, so we didn't want to make the same mistake twice. So the two strands being complementary meant that the information necessary to reconstruct one was present in the other. But again, just because it makes sense doesn't mean it's right. And so they kind of brainstormed these alternatives. And so they're like, well, it's possible that the molecule pulls itself apart, copies it, but both of the daughter strands actually stick together. And the two original parent strands are also sticking together. It's possible that we pull it apart and a daughter strand goes with each one. We now know this to be the case. And then we also have what's called the dispersive model. I like to call it the chunky model, which is like a hybrid between this one and this one, where different chunks go into different places where just everything gets distributed. Okay, we make copy and everything gets distributed. Okay, I don't really know if there was ever a mechanism for this that was suggested because it's kind of fuzzy to me, but that was one of the ideas. So along came Messelson and Stahl in the 1950s. It was a very busy decade for the DNA molecule. And they came up with a pretty clever experiment, and it goes back to using labeled molecules. So they grew bacteria in heavy nitrogen, right? Nitrogen with an extra neutron. Bacteria, remember, grow very, very quickly and reproduce very, very quickly. E. coli usually, if they're warm at the right temperature, will divide about every half an hour. Okay, it could be faster or slower, but you can figure it out, right? How long does it take for this guy to divide? And so they grew all these bacteria in heavy nitrogen, which means remember this nitrogenous base on that molecule, so all of the nitrogenous bases are heavy, they're marked. So then they take these guys, they take them out of the heavy nitrogen and they put them in light nitrogen, and they let it replicate, they let the bacteria divide once, just once. They pull out a sample, they let them divide a second time, pull out another sample. So then they're looking, they take that DNA, they centrifuge it down, and they're looking to see, is all the DNA the same, or is some of it different? And sure enough, this is separating again, this was centrifuge to separate stuff based on mass or density. So we had this one band in the middle, and that debunked this idea. Because we knew that we had, divided our bacteria, and so we had replicated our DNA, and the fact that we had all of the DNA was the same density meant that we did not have the conservative model going on. So if we had the conservative model going on, we would have had some heavy and some light. It's not what happened. But the semi-conservative model was still kind of being contended by the chunky model, which is what I call it. So then, they took their second replication, spun it down, and got this. They had a band that was less dense and a band that was more dense. Well, that's how we kind of debunked this chunky model idea. Because we would have only had one band that would have progressively gotten lighter and lighter and lighter if this is what was happening. But that's not what we got. Instead, we had two bands, one that was the same density as the first one, and then one that was all light. So that's how we settled on semi-conservative model being the way to go. So the basic principle of DNA replication is very simple. You pull the two strands apart, use each as a template to generate a new strand. But the details are very, very complicated. And so we try to use simple organisms, bacteria, to understand this process. I want you to bear in mind that you have about 6 billion nucleotide pairs in each nucleus. 6 billion. If we were to type them out in a book, it would take 1,200 books the size of your textbook to produce how much DNA is in one nucleus. That is a stunning amount of DNA. But your cells are able to replicate that DNA during the S phase of cell cycle in a couple of hours. That's typing really fast. And then the error rate is 1 in 10 billion nucleotides. That's insane. How does it manage this? And what we find is actually because we can do this artificially now, nothing that we can design is anywhere near as accurate as what your cells do themselves. Nowhere near as accurate. And we'll look at that in Unit 5, actually. So to get started, replication begins as what we call the origin of replication. It moves outward in both directions from that origin. Bacteria, remember, have one circular chromosome, so they have one origin of replication, and literally, at that origin, the two strands are pulled apart and the new daughter strands form in the middle, and then they get pulled further and further and further, and the daughter continues to fill in until they split. Remember, DNA needs to be double-stranded. So we don't pull them apart and then add, we do it a little bit at a time. Because DNA is only stable when it's got that double strand. Eukaryotes, of course, have the multiple linear chromosomes, rather, but multiple origins of replication, and the same idea works. Pinch it apart, fill in the middle, and then these, we call them bubbles, move toward each other until finally they meet, and we end up with 2 strands. It's in general how it works. It's been my experience that if I take about 4 minutes and I show you a video, then our next 10 slides will go much better. So we're going to do that. So this is the BioFlix video that you guys are going to watch on your mastering. So of course you could skip it when you get to it mastering. If you do it, like tomorrow, you'll probably remember. I'm going to fast forward to here.
Every cell in your body is produced by cell division. Before each cell divides, it must copy its genetic material in a process called DNA replication. Understanding of DNA replication comes largely from studies of E. coli, bacteria that are found by the billions in your large intestine. Let's take a look at how DNA replication occurs in an E. coli cell. As we zoom in, we see the DNA. At the origin of replication, the 2 strands of DNA separate, serving as templates for making new strands. The result is a replication bubble. The bubble grows in both directions, forming 2 replication forks. Let's zoom in on one of them. Many proteins work together at the replication fork. Only some are shown. Here, the DNA is unwound, and DNA polymerases, shown in orange, build new strands of DNA. Original parental DNA strands are shown in dark blue. Newly formed DNA strands are shown in light blue. Because strands in a DNA double helix run in opposite directions, the new strands must be made in different ways. One new strand, the leading strand, is built continuously. The other new strand, the lagging strand, is built in pieces. First, let's focus on the leading strand. DNA polymerase builds a new strand of DNA by adding DNA nucleotides one at a time. Each new nucleotide must pair up with its complementary nucleotide on the parental strand. Adding new nucleotides works the same way on both the leading and lagging strands. Each piece of the lagging strand begins with a short segment of RNA, shown in red. surrounds the RNA and attaches to DNA polymerase, which builds the rest of the new piece as DNA. When the piece is finished, it is released from DNA polymerase. How are pieces of the lagging strand joined together? A different DNA polymerase removes RNA and replaces it with DNA. However, it cannot finish connecting the pieces. An enzyme called DNA ligase joins the pieces together. Growth of the leading and lagging strands continues on both sides of the replication bubble until there are two identical DNA molecules. Although bacteria are very different from humans, the process of DNA replication in bacteria is similar to what happens in your own cells.
Oh, thanks. All right, so let me go back to where we just came. I need to move that slide. There we go. So in essence, we talked about this a second ago. In essence, the information for a duplicate copy of DNA is held on both strands. And so what we do is we pull them apart and then we fill in the opposite side. That's hard to do, it turns out. And it's hard to do because of the chemistry of this. First of all, remember that DNA is a very stable, long-living molecule. And how is it stable? It's stable when it has another strand, and it's stable when it's all twisted up. That's why we call it that, that alpha helix. So in order to pull them apart, you have to make it chemically unstable. It doesn't like that, right? Because that's going to make it, well, chemically mad. So what do? Well, when we pull them apart at that origin of replication, we have a protein that comes in and binds to it so that it doesn't get mad. Because remember, what it should do is just go snap back together, pull it apart, snap back together. So our single-stranded binding protein holds them apart. It's this protein called helicase that's actually pulling the two strands apart in advance of the single-stranded binding protein, keeping them that way. But then we also have this issue of the twist. Remember, the molecule is twisted. And so as we do this, we have to untwist it. That adds a lot of pressure to both ends of this replication fork. So we have this thing called topoisomerase. Topoisomerase relieves the pressure, literal physical pressure that's building up on that molecule, because what we don't want is to snap the backbone in half. It's just a covalent bond, right? We don't want to break it, which we could do. And so the topoisomerase relieves some of that strain. So we also have something kind of annoying that has to do with DNA as well, has to do with DNA's chemistry. The nucleotides themselves, the individual monomers themselves, can only be attached to the 3' carbon of a previous nucleic acid. So remember when we learned this? I mean, did you guys learn your carbons? This was 1, 2, 3, 4, 5. Okay, 5 was attached to your phosphate. 3 was kind of down here. That's your 3' end. That's why we call it the 3' is because that's your 3 carbon. Okay. We can only attach a nucleotide to a 3' end, which means this molecule can only grow in this way. Okay. And then the complementary molecule could only grow, like if we had its other friend over here, it could only grow in that way. So the chemistry says we can only add to the 3' end. Functionally, this means two things. One, a DNA nucleotide, a monomer, is not going to attach to nothing. They're very, very, very picky. So that's a problem. And then, once you have a starter, you can only attach to one side of it. Also very annoying. So we use RNA to help us with this. Remember, RNA is much more flexible. RNA doesn't care. It's a short-lived molecule. It comes and goes and all this sorts. RNA is like the cool cousin. Meh, whatever, I throw my hands in the air. So we use RNA to be a primer. Primer means first. So RNA comes in first and attaches, because RNA will attach to anything, it doesn't care. RNA is going to attach here, and it will expose its 3' carbon. And once it does that, then the DNA is like, oh, okay. DNA just goes. Good to go. Same thing happens over here. So remember, we pulled apart our replication fork. We built an RNA primer here, and added onto our 3' end over and over and over and over. The problem is this part, because we can't grow like that. We can't do that. We can't go this way. So this strand is only going to grow like this, and this strand is only going to grow like this. And so as we pull this apart, we keep adding little RNA primers and just building these baby chunks. Baby chunk, baby chunk. We call these Okazaki fragments. That's the guy that figured it out. And these are just the exact same thing as this, only small. And so this is what it's saying, that we have that lagging strand where we build our RNA primer and we fill that in. And then the whole complex of molecules comes here, build that RNA primer, fill this in, here, fill it in. And it just jumps backwards. It's like I go forward 2 steps, and then I go backwards 4 steps. And then I go forward 2 steps, and then I go backwards 4 steps. As long as I'm always walking in the same direction, we're good to go. But I have to do it in little big chunks, otherwise it doesn't work. So the leading strand, like I said, is very easy. A few more proteins that you guys need to know. We have primase, primer, very easy, RNA primase. It comes in, we build our primer, so that allows our DNA to just be added to the end of it in that 3' direction, no big deal. As it gets here, the fork gets bigger and bigger and bigger because we have helicates unwinding it. Topoisomerase is over here absorbing the pressure. Single-stranded binding protein is down here, no problem. The lagging strand is much more complicated. Okay? The lagging strand, like I said, first we build our primer, and then our polymerase comes in and builds our DNA, and then very rapidly, it only builds a small chunk, but it gets to the previous RNA primer. And then this comes out, and we move backwards, right? And we build number 2. We build number 2. And then we have this other issue. We have to get rid of the RNA. We're not trying to build an RNA molecule. We're trying to build a DNA molecule. And the RNA is not stable enough to last years. So then along comes another DNA polymerase. The first DNA polymerase was named DNA polymerase 3. Oops, where is he? Here. Okay. This is called DNA polymerase number 1. It's like with the photosystems. Who does this? All numbered wrong. DNA polymerase comes in. Number 1 comes in and removes that RNA primer. The last thing that needs to be done is to build that last covalent bond. Ligase comes in to do that. In both of the strands, this is what I was just saying, we need to remove the primer, weld the DNA together. DNA polymerase 1 removes the RNA primers, and the ligase builds that bond. This slide puts it all together very nicely. This is the level of detail you need to know. It is annotated perfectly in your textbook. It is available unlabeled in your notes questions. So post-it note it and then compare it to your textbook to make sure that you've done it properly. Okay? And that's everything that we just discussed. So like we said at the beginning, this process is extraordinarily accurate, but it is not perfect. Told you before, we had errors at about a rate of about 1 in a billion. Interestingly though, the pairing errors between the incoming nucleotides and the template is about 1 in 100,000. That's a very different number than 1 in a billion. Okay? Well, how do we fix that? During replication, it turns out those DNA polymerases actually proofread the nucleotide against the template as it's added. So you know how when you're typing on a keyboard on a phone, and you make a typo, and you have backspaced it before you even realize that you backspaced it? It's like you're typing and you correct your work as you type it. And it doesn't even, and you're so good at typing, you don't even think about it, you just do it. That's what's happening here. Because remember, this is happening insanely fast. And so the typos are corrected literally by the polymerase as soon as they happen, most of the time. If typos still exist after that, there are a whole host of repair enzymes that come along and proofread for editing. And if an incorrect nucleotide is found, it's removed and replaced. you can have errors that arise after replication. DNA can be damaged, UV rays, carcinogens, things like that. What kind of an error could that be? I'm going to show you because this is kind of a weird thing to think about. When you add it, this is a UV error. UV rays, a lot of energy comes in, and look at what happens to those two bases. They actually bind to each other instead of binding to their opposite one. So it's an example of an error that could be found. We have to fix this. If there's a mismatch error, as in the DNA is put in a wrong one and that's not caught, we call that a mutation. Okay, we had a mutation during cell division. That's why most of the things that cause cancer are things that are causing an increased rate of cell division. If your cells are dividing all the time, you have an increased chance of having an error that is not caught. Right? So that's why things that cause cancer, things like sunburns, is what happens when you burn. All your skin peels off. They have to divide more. Right? That's what's happening there. Most mutations are problematic. So it's not a surprise that we have all of these different DNA repair enzymes. In fact, there's 130 of them that we know of present in humans, that their whole job is to just look at DNA after it's been produced. Remember when we talked about the checkpoints? When we talked about cell cycle checkpoints? And I said, what's the S checkpoint looking for? Looking to see that your chromosomes were duplicated properly. Okay, that's where these are all coming into play. Okay, so what happens when we find an error? Well, we have, like I said, there's like 130 different ones. We're going to just generally call it a nuclease. They all have names that you don't need to know. And the nuclease comes in, removes the chunk. Our DNA polymerase comes in. It's the same enzyme that we use to build the DNA molecule in the first place. Comes on in, adds our DNA monomers. We don't need RNA here because we had DNA there, so we don't need to put a primer in. And then the ligase comes in and seals the deal. I want you to consider one more problem for linear chromosomes, and that is the ends of the chromosome. So if this is my chromosome, and I go through replication, well, normally I end up with a little bit of primer here, RNA primer, that's where my DNA comes in and attaches to. And then later I come in and I remove this RNA primer once I've built my next molecule out here. But on the end of my chromosome, there's nothing for the DNA to attach to. I can put my RNA there, but then the RNA is going to fall off and I don't have any other DNA there. So what do you do about that? Well, nothing. It turns out every time your cells divide, your DNA replicates, you lose an end, a little bit off the end, every single time you replicate. Okay? And so chromosomes have these little protective ends. We call them telomeres. They look like little stocking caps in the picture. They don't actually. And all they are extra DNA. They don't code for anything. We don't call it junk DNA because junk DNA isn't real. But they're these protective ends that don't do anything. So that every time your DNA duplicates, we are going to lose a small piece. We know that. But the piece that's lost isn't damaging to gene expression. So it's believed that these shortening, so that over time they get shorter, shorter, and shorter, actually can lead a cell to undergo apoptosis. What's apoptosis? Cell death. So if a cell runs out of telomeres, it's going to program cell death. Why? Because we don't want to eat into the coding area. We would never want to erode away this chunk of the DNA because then we won't be able to produce the proteins that we need. So that makes sense, but remember that your chromosomes came from your parents, quite literally, right? We said that, that's where they came from. So if your parents don't have any telomeres, how do you get them? Because your parents' telomeres are getting shorter and shorter and shorter as they divide, so how do you have telomeres? Well, it turns out there's an enzyme called telomerase, and telomerase is active in germ cells. Germ cells are the cells that become eggs and sperm. So what telomerase does is it makes sure every single one of those eggs and sperm have chromosomes with a full telomere at the end. Okay, that's the job of a telomerase. Well, that's all well and good, and perfect, actually, because you would want to have a full complement of telomeres. However, so here's your telomeres, somebody tagged them, look how cute they are. we're finding that telomeres are actually active in cancer cells. And that would make sense, right? Because what's cancer? Uncontrolled cell growth. We don't want the cell to undergo apoptosis. We want the cancer cell, would want to just keep dividing indefinitely. So a lot of research going on right now with regard to telomerase and figuring out how to disable it in a cancer cell so that if it keeps dividing, ultimately it would go into apoptosis. It's just an area of research. Like, okay, how can we turn off this telomerase? Because we need it to stop working. So our last short topic here today is about how do we package our chromosomes. So I told you this earlier, the DNA in your chromosomes would be about 4 centimeters long stretched out, and you've got 46 of them. That's a lot of DNA to be held in a nucleus. We have to condense it and package it in order to make it fit. In a bacteria, remember bacteria have one single circular chromosome, and that part is it's coiled in and on top of itself in order to pack it very, very densely. We call that the nucleoid region in a bacteria because it's not contained in a nucleus because there's no internal membranes there, but it's just this region within the bacteria. So this is what it looks like. That's one circular molecule, which is kind of crazy to see, but that's what it looks like. Eukaryotes, however, are multiple linear chromosomes, and so each one is one molecule associated with a whole lot of protein. That's the protein that got us in trouble at the beginning of our lecture. Well, it turns out what we do with our DNA is that first we wrap it around these proteins called histones, and the histones have these tails. The tails matter later. So we wrap the DNA around our histones, and then the histones fit on top of each other kind of like Legos, so they fit together very tightly. And then these form loops, and then the loops spin to become a chromatid. Now remember, we only actually look like this when we are dividing. Okay, we don't normally look like this. And the reason is because the DNA has a purpose, and that purpose is transcription and translation for the purpose of protein synthesis. Well, in order to undergo transcription and translation, which is what we're going to start next week, we have to be able to access the DNA molecule. You have to be able to get to the DNA. So this is DNA I can access. This is DNA not so much. I can't get to this. So when we look at the levels of packaging, the regions of the chromosome that are being expressed are more spread out, and the regions of the chromosome that are not being expressed are more condensed. That's what we end up having to do. So, and we can go back and forth, right? Because some genes are active during some periods of your life and then turned off later and whatnot. During interphase, most chromatin is in the 30 or the 10 fiber stage and some looped domains. So during interphase, it could be any of this. Okay, any of this right here. Okay, that type of chromatin is called heterochromatin, so different chromatin. Could be any of it. We also have something called euchromatin, true chromatin, which is more dispersed, and so it looks more like this. So here's euchromatin, I can get to the DNA here. Okay, heterochromatin is compact, I can't get to it nearly as much. Okay, that's what we've got there. The looser packing of chromatin here makes it accessible. So how do we change this? We change what's on those tails. Okay? We can add different functional groups to these tails to either make them repel each other or be attracted to each other. So we don't change the DNA, we change what's happening to those histones to cause them to move apart or come together. So this is what it ends up looking like. Okay? Where this reversible chemical modifications allow it to go back and forth between those different levels of condensation. This is our last slide. This is not a video. It's just a screenshot. It's on YouTube because when you're a science teacher, all sorts of interesting things pop up on YouTube. This is a nucleus. This is a model of a nucleus with 46 chromosomes. You know how I always tell you they're bowl of spaghetti chromosomes? So here she color-coded all of the different chromosomes so that you could see just what a mess it is. And then within that, some of it, I'm going to turn it off because it's actually easier to see like this. And then within that, you can see they're all tangled in a gigantic mess. And you can really appreciate the importance of the super condensing into that X structure before mitosis. Imagine what would happen if that went into mitosis. Do you think we would tangle and break? Probably, right? Probably we would. And so if that's what our nucleus is really looking like during interphase, we would really need to super condense into those nice little compact X's that are easy to move around on our microtubules. Okay? And so anyway, I thought that was just kind of a fun image to illustrate that.